Since the late 19th century, communication technologies for transmitting information over distances have been revolutionizing the way humans, sensors, and robots communicate. Different from wired and wireless communications, molecular communication was proposed to transmit chemical signals between human, cells, and the environment, typically at the micrometer or nanometer scale, by mimicking the biochemical means of communication developed by the nature. To unleash the potential of molecular communication for biocompatible micro and nanoscale applications, it is critical to develop chemical signal processing blocks in the molecular domain, thus ensuring the biocompatibility that the use of electronic devices would prevent. To address this challenge, we developed a microfluidic molecular communication (MMIC) platform to investigate the possibility of processing signals via chemical reactions. Our MMIC platform consists of a transmitter that encodes the signal into the concentration of the chemical, a microfluidic propagation channel, and a receiver that decodes and detects the signal. Through the joint design of a selection of chemical reactions in microfluidic geometry, our MMIC platform achieves three chemical signal processing functions: signal shaping, signal thresholding, and signal amplification. Chemicals are injected into the transmitter through our 3D printed syringe pumps, which are controlled by Arduino boards connected to a computer. All pumps are actuated simultaneously, and the interaction between chemicals is achieved by adjusting the tubing length. At the other end of the propagation channel, the receiver is also using 3D printed pumps to inject the chemicals required for the thresholding and amplifying functions. The pumps are operated by a different computer, thus making the transmitter and the receiver independent of each other. In our platform, we selected an ultraviolet visible spectrometer as the detector due to its capacity to detect and identify a large selection of molecules. To achieve the designed signal processing function, we carefully selected the chemicals and prepared all the solutions in our chemistry lab. Once prepared, the solutions were loaded into standard plastic syringes and installed inside the syringe pump for injection into the transmitter and the receiver. The MMIC platform is controlled by our open-source Python software. Through a graphic user interface, the software translates the user settings into a series of commands to control the hardware and collect the data measured by the flow meters and the spectrometer over time. As proof of concept, we encoded the signal Y in the concentration of sodium hydroxide and represented the bit 0 and bit 1 of a message using two different concentrations. Before being emitted at the output of the transmitter, the signal Y is processed through a reaction within the solution P, prepared here with hydrochloric acid. This reaction leads to a consumption of Y and result in a complete signal suppression. The computer actuates the syringe pumps containing the different solutions and inject them simultaneously into the transmitter. Y and P are both paired with a pump injecting a solvent to ensure a constant flow rate at the transmitter output all the time. Through the reaction between Y and P, the transmitter will emit a pulse-shaped signal Y. The width of the emitted signal Y can be controlled by adjusting the tubing length of P and delaying its interaction with Y. This pulse-shaped signal Y then leaves the transmitter and travels through a 25-meter-long propagation channel. The receiver of our platform is designed to process and decode incoming signals using different chemical signal processing. One of the main requirements for decoding the information carried by the chemical signal is that the receiver output should be visible and quantifiable by the spectrometer. However, the solution Y does not fulfill this requirement since it is a colorless solution invisible at the spectrometer. To solve this problem and demonstrate the versatility of our platform, we designed a chemical reaction between Y and a solution AMP for the receiver. This solution was made of bromothymol blue, a pH-sensitive dye that can react with the sodium hydroxide of solution Y to change its color, switching from a yellow acid form at low pH to a blue base form at high pH. This blue base form, named the receiver output signal O, can be detected by the spectrometer. We also designed at the receiver a thresholding reaction between the incoming signal Y and a solution of hydrochloric acid. This reaction reduces the noise and interference introduced by the propagation channel. By consuming a fixed quantity of the chemical signal Y, This reaction also selects the minimum amount of Y that can be detected by the receiver. In addition to allow the detection of the incoming signal, the AMP solution also acts as an amplification reaction. It amplifies the receiver output signal O to a level high enough to be detected, and ensures that in the presence of Y, the concentration value of O is not influenced by the concentration of input Y. With the thresholding and amplification signal processing functions, we are able to decode the invisible signal Y and measure it using the spectrometer connected to a computer. We used this setup to transmit a simple message from the transmitter to the receiver of our platform. The characters of the message were converted into bits using the ASCII encoding. Each bit 1 was transmitted by an injection of solution Y, with a rectangular profile defined by the bit interval and duty cycle. While for bit 0, only the solvent pumps were left running to generate a baseline. To indicate the start of the message to the receiver and specify the duty cycle and bit interval used for the message encoding, 2 extra bit 1 were also added and transmitted at the beginning of the information bit sequence. The signal was successfully measured at the receiver and decoded to restore the message. We tested transmission at different data rates and were able to maintain a good communication reliability via adjusting the chemical reaction settings. Our MIMIC platform illustrates our methodology for designing, optimizing, and validating diverse signal processing functions over the molecular domain based on chemical reactions. Most importantly, by replacing the species with the application-specific chemicals and shrinking the hardware into a compact, light, and portable lab-on-a-chip device, our MC platform is also a major step toward the promised MC applications in the fields of medicine or entertainment.